So picking up where we left off with the last lecture, talking about the prokaryotes, bacteria and archaea, we've covered what they are. We've talked a little bit about why we should care about them, and we talked about when they evolved. And today we're going to spend some time talking about why they're so successful, specifically their metabolic diversity, and that they're the masters of evolution, and why they have so many unique traits and the ability to evolve so many unique traits. Before we get into that, though, I want to start off with a clicker question. So this is just a review from the previous lecture. Which of the following is a true statement? Okay, if you said C, you are correct. Prokaryotes were the first forms of life that appear in the fossil record approximately 3.5 billion years ago. A is false because it says archaea are more closely related to bacteria than eukaryotes. When we actually look at the phylogenetic tree, archaea, although they share many traits similar to bacteria and hence they're grouped in this paraphyletic group, the prokaryotes, the archaea are in fact more closely related to the eukaryotes. Archaea have similar traits to bacteria, that is true, but linear DNA is not one of those traits. Their DNA is circular. And they do not have organelles. They don't have a nucleus. That's a trait that's unique to the eukaryotes. But they can do asexual reproduction. Most bacteria are not, in fact, bad for human health. Most are either good or have no real effect on human health. It's just a few bacteria that really negatively affect human health. So picking up where we left off talking about bacteria and archaea, the prokaryotes, and why we should care about them, we addressed their metabolic diversity, or we talked a little bit about their metabolic diversity, and we're going to talk today a little bit about that in a little bit more detail today. So when we're talking about metabolisms and the nutritional needs of all organisms, there are two fundamental things that all organisms need. The first thing is an energy source to make ATP. And remember, ATP is sort of the functional currency of the cell and cellular level reactions. They need ATP to do all the stuff that cells need to do. And typically, the energy source to make this ATP, to make the cellular currency, is from what's called the oxidation of reduced carbon molecules. So when we talk about reduced carbon molecules, we're talking about high energy carbon molecules, things like sugars. And if they're not getting that energy source from high energy reduced carbon molecules like sugars, they're getting them from sunlight. So when we're talking about the production of ATP from reduced carbon molecules like sugars, typically what you guys will think of is aerobic cellular respiration that hopefully you've learned about either in high school or in previous lectures here at JMU. And I'm not going to get into all of the details of cellular respiration and how it works. Just know that this is a cellular process that happens in the mitochondria of eukaryotic cells, where they take this high energy sugar molecule, glucose, and use the chemical energy that's trapped in that molecule to produce lots of different ATPs. And that ATP, once again, is the ultimate energy source that those cells are going to use for cellular level reactions. So you're taking the energy in a reduced carbon molecule like sugar and transferring it into the energy in the molecules or in the molecular bonds of ATP. Alternatively, some organisms use sunlight to produce ATP or other high energy molecules like NADPH. And in this case, you have energy in light getting converted into chemical energy in the bonds of NADPH and ATP. The second thing that's a fundamental nutritional need of all organisms is a carbon source for the synthesis of carbon-to-carbon bonds in the organic molecules that make you and I and all organisms up. So remember, we are carbon-based organisms, and in fact, all organisms are carbon-based organisms. And if you're going to build an organism out of carbon molecules, you need to get those carbon atoms from somewhere to build those different organic compounds that make us and all organisms up. So things like DNA, things like RNA, things like proteins, things like sugars, things like lipids, those all have to come from carbon and built from carbon from the ground up. And so there's two major places that organisms can get carbon molecules from. One is from organic sources. In other words, we get those carbon molecules from other organisms. So we can get sugars, fats, proteins if we eat another organism, whether it's a plant or an animal or anything that used to be living at some point, even a mushroom. We're taking their organic carbon molecules. When I say organic, it means it comes from a living organism, and converting them into the carbon molecules and the organic molecules that make us up, or whatever organisms, what are eating whatever organism. Alternatively, some organisms can get a carbon source from simple inorganic molecules. So when I say inorganic molecules, what I'm talking about are molecules that are not from living organisms. So the classic example of this would be the Calvin cycle in photosynthesis, that once again, I wouldn't expect you to memorize all of this, but hopefully you remember at some point learning about the Calvin cycle. And the big job of the Calvin cycle is to take CO2, which is an inorganic molecule, and run it through this Calvin cycle to make this organic molecule called G3P, which ultimately gets turned into glucose and fructose. So basically you have CO2, which is not like a highly useful molecule in terms of being biologically reactive, and converting it into this G3P molecule, which is a very biologically useful and reactive reduced molecule and a great carbon source for synthesizing carbon to carbon bonds. So if you take all of this and you break it down into this sort of matrix here where you've got different sources of energy for ATP synthesis, and you've got different sources of carbon to carbon bonds for biosynthesis, or making up biological molecules that are made of carbon. You can break organisms down into these two sort of types of trophs. So you've got phototrophs that feed on light to make ATP, and you've got chemotrophs, or chemical feeders, that feed on chemicals to produce ATP, or sugars to produce ATP. And then in the source of carbon to carbon bonds, you can have autotrophs that are self-feeders. These are essentially organisms that use inorganic molecules, for example, CO2, to make carbon to carbon bioreactive molecules. Or you can have heterotrophs, that translates to other feeders, that basically use organic molecules from other organisms as their major source of carbon for making carbon to carbon bonds. Okay, so when you combine these things together, you can get the different types of organisms and the metabolic types that we see out in nature. So for example, you can have photoautotrophs that use light as an energy source and inorganic molecules as their carbon source. Or you can have chemoautotrophs, or photoheterotrophs, or chemoheterotrophs. So you can break all these organisms or different organisms down into these four major groups. Okay, so given this, which plant or which box would plants go into? A, photoautotrophs, B, chemoautotrophs, C, photoheterotrophs, or D, chemoheterotrophs? And the second question is, which box would animals go into? So go ahead and answer that question. So if you said A, plants go into A, they are photoautotrophs, you are correct. They get their energy from light, and they get their carbon source from CO2. If you said animals are chemoheterotrophs, you are right. There are very, very few rare, weird exceptions in the animal world that are photoheterotrophs, but 99.999% of animals are considered to be chemoheterotrophs, where they get their energy by eating organic, or they get their carbon source from eating organic molecules, and they need high energy chemicals or sugars to produce ATP. Now, interestingly, bacteria, where do you think they go? Bacteria and archaea. Perhaps not surprisingly, they can do all of these things. So that doesn't mean that one type of bacteria can do all of them. It means different species of bacteria have evolved different types of metabolism. So some bacteria can do chemoautotrophy, some can do photoheterotrophy, some can be chemoheterotrophs, some are photoautotrophs. But across the board, they are the most metabolically diverse group of organisms because they can use either light or inorganic or organic sources for energy and for carbon sources. So all other organisms pretty much are either phototrophs, photoautotrophs, or chemoheterotrophs, whereas bacteria and archaea can be any of these four types of trophs or practice any of these 4 types of trophy. And once again, that doesn't mean all bacteria can do all 4 types or an individual bacterium can do all 4 types. It means across all the different species of bacteria and archaea that we see, some of them, or some individual species can do this, some individual species can do this, some individual species can do this, some individual species can do this. So how do they do these different metabolic activities? So if we look at metabolic diversity in prokaryotes, actually let's start off not with prokaryotes, but in more familiar organisms and look at how they do cellular respiration. Okay, so when we think of cellular respiration, typically we think about the cellular respiration that we learned about in high school or Bio 140. And essentially what you're thinking of is aerobic cellular respiration. So when I say aerobic cellular respiration, what I'm talking about is cellular respiration that consumes O2. So it requires O2 to proceed. And this is the equation for aerobic cellular respiration, the chemical equation for what happens, where you have a glucose molecule that's a high energy reduced molecule. You have oxygen that's an extremely electronegative molecule. And that glucose and the carbon in it gets oxidized to CO2, whereas the oxygen gets reduced to H2O. So remember, oxidation is a loss of electrons, and what that means is typically you're losing hydrogen atoms, and so you have the carbon molecule being stripped of its hydrogen atoms and losing some energy associated with those bonds, and then you have an oxygen molecule being reduced to H2O. And when we talk about reduced, reduction is a gain of electrons, and typically you're gaining hydrogen atoms with that too. And so you have this oxygen gaining hydrogen atoms. And as a result of this process of aerobic cellular respiration, the chemical energy that was trapped in the chemical bonds of this glucose molecule gets converted into energy to reduce ADP into ATP. So you take some of that energy that was trapped in these bonds, convert it into the energy of those bonds to produce ATP. That's aerobic cellular respiration. When we're doing aerobic cellular respiration, sugar is consumed and oxygen is taken in, okay? So the reactants in this equation require sugar and require oxygen. And in this equation, the sugar, or the carbon, highly reduced carbon molecule, is an electron donor. It's donating electrons and donating energy, whereas oxygen in this case is an electron acceptor. It's a lower energy molecule that's accepting electrons. And in order for these sorts of reactions to occur, you need to have an electron donor and an electron acceptor. This is something called a reduction oxidation reaction, or a redox reaction. A redox reaction is just one molecule is being oxidized, another molecule is being reduced. So one molecule is losing energy when it's being oxidized, another molecule is gaining energy when it's being reduced. And anytime that you have a reaction where one molecule is being oxidized, you need another molecule there to balance the equation and accept those electrons to be reduced. So these are called redox reactions, and they're necessary for these metabolic processes that go on in organisms. So what do organisms do if there's no sugar or no oxygen around? How do you do cellular respiration, or how do you have a metabolism if oxygen and glucose, these electron donors and acceptors, aren't available? Well, prokaryotes, humans, obviously we can't do that. We need oxygen to survive. But prokaryotes can actually use a bunch of different electron donors and different electron acceptor molecules to make ATP that they need. And this, or an example of this, would be anaerobic cellular respiration. So cellular respiration that does not require oxygen. And that's one metabolic process that prokaryotes have evolved to do. So let's look at this another way, where we're looking at normal aerobic cellular respiration. So this is what plants and animals do, where you have C6H12O6, glucose. That's the electron donor molecule. This is the high energy molecule that donates electrons and energy. And then you have O2, which is the electron acceptor molecule. And then you have these byproducts of the reaction, which are CO2 and H2O. In addition, this produces some ATP. So this is typical normal aerobic cellular respiration that we see in plants and animals. So let's look at what bacteria and archaea can do. sorry, this is another way of looking at this where you have the electron donor, which is sugars, and the electron acceptor, which is O2. So this is just that same equation again. This is aerobic cellular respiration that we see in eukaryotes. When we look at prokaryotes, they can use a wide variety of other molecules as electron donors and acceptors to do cellular respiration. So this is still considered cellular respiration. It's just oftentimes it's not aerobic. So they can use a bunch of different molecules as electron donors, not just organic sugars, but they can also use methane, hydrogen sulfide, nitrate, nitrite, lots of different types of molecules as electron donors, and lots of other molecules other than oxygen as electron acceptors. And in fact, if you look at these equations, or I'm sorry, these electron donors and acceptor molecules that are outlined in these blue boxes, in these cases, there's no oxygen required. And so basically, these prokaryotes are able to do cellular respiration without oxygen because they can use these other molecules as electron acceptors instead of oxygen. And once again, I want to repeat that typically each prokaryote species can only do one of these individual reactions. So only one of these reactions, or only this reaction, or only this reaction. They can't do, one species can't do all of them. So if we look at this a little bit further, so before we were looking at sort of the reactants that are used to do cellular respiration and to do these sort of metabolic activities to produce ATP. Now let's look at the byproducts in this reaction. So if we're looking at normal cellular respiration, the normal byproducts are CO2 and H2O, and then we get some ATP as well. And if we look at this another way, with normal aerobic cellular respiration, you have the electron donor, you have the electron acceptor, and then you have these byproducts, CO2 and H2O. So let's look at what byproducts we can get from these different prokaryotic forms of cellular respiration. So these are those same different reactions that bacteria and archaea can do. And here we're looking at the electron donors and the electron acceptors. And here we're looking at the byproducts. And so you can see that normally we get CO2 or H2O when we're thinking about the cellular respiration that plants or animals do. And you can still have H2O as a byproduct in many cases. You can also have other weird molecules, so things like methane or hydrogen sulfide or iron or nitrates or nitrites. And the thing that I want to point out here is oftentimes the byproduct of one organism's cellular respiration. So let's say you have one bacteria that can do this reaction or requires these reactants, and it will produce this SO4 sulfate as a byproduct. So this byproduct here can actually be a reactant in the metabolism of a different species that's able to do this reaction. So sometimes byproducts can be reactants. And this diversity enables sort of a recycling of those byproducts. And we see that not just in the sulfates, but also in the nitrates and also in the nitrites. And so there's lots of different byproducts that are trash to one thing that are treasure to another thing in terms of being a food source. So why would it be advantageous for a species to have these diverse metabolisms and use so many different electron acceptors and donors? So why have bacteria evolved all these different types of metabolisms? Think about that for a sec. What might be one advantage? So one is this allows bacteria and archaea to live in crazy environments where other organisms can't, specifically because they're able to survive in areas where there's no oxygen and there aren't what we would consider normal food sources, but there might be a lot of sulfur or methane or other types of molecules that are useful to them metabolically speaking. So they can live off molecules that other organisms can't, and that will overall reduce competition. So they're able to eat things that nothing else can. Nothing else can survive there, so it's just a feast for them and no one else. And so it's much easier for them to survive. It lowers competition. And it also allows them to coexist with other prokaryotic species or eukaryotic species by cleaning up each other's messes. So in other words, things that are junk byproducts for one organism's metabolism is food for another organism. And once again, you're reducing competition for resources. They can survive on things that other things wouldn't want to eat, and that reduces overall competition and gives them a competitive advantage. So let's look at an example of this. Specifically, let's look at these things called microbial mats. So this is sort of a big sort of salt marsh where you're just looking at sand and it's just really, really salty water with not a lot of nutrients in it that not a lot of things can survive in. And if we sort of dig down a few layers in these microbial mats, we can see that there's different layers of microbes. So you can see this green layer, this purple layer, this kind of gray layer, and this black layer. And those layers represent different species or types of microbes that have different metabolic capabilities. So if we break these sort of layers that you're looking at here down in this figure right here, you can see the depth beneath the surface. So we're going from the surface all the way down to the bottom layer of the sand. And we can see first how deep light penetrates. So light goes down about 3 millimeters. And then we can see the penetration of oxygen. So oxygen goes down about 2 millimeters. And so this is the availability of light in these microbial mats, and this is the availability of oxygen in these microbial mats. And next, what we can do is actually measure the concentration of O2 and H2S. So these are two potentially important molecules for these different metabolic processes. So remember, oxygen is important for aerobic respiration, for example. And so we can see that oxygen levels kind of spike here and then go down, and then we see this H2S levels spiking as we go further down. So increases in the availability of that molecule. And from this, we can actually look at the types of metabolisms that exist in these different layers based on the availability of these different resources. So we can look at autotrophic groups of metabolisms and heterotrophic groups of metabolisms. If we start off looking at the autotrophic group of metabolisms, you can see that in this top layer here, we have oxygenic photosynthesis going on. So why oxygenic photosynthesis going on here? Because number one, we have oxygen available, and number two, for photosynthesis, we have light. So oxygenic photosynthesis can produce energy here, and that's the type of metabolism that's being used here because there's light and oxygen available. In addition, for the heterotrophic metabolism, we have aerobic respiration. So this is the typical sort of cellular respiration that we think of because there is oxygen available. And so we have two different major metabolisms happening here in the layer that has oxygen and light available. Now let's look at the layer here where we go down a little bit further past the 2 millimeter mark, and all of a sudden we're into an area where there's no longer oxygen, but there is still light. And so in this area here, we have organisms that are capable of doing anoxygenic photosynthesis. So they can do photosynthesis, but they don't need oxygen. And so it's a special type of photosynthesis that doesn't require oxygen. And then we can look in the areas where there's no light available and there's no oxygen. And in that case, we have bacteria and archaea that are doing chemoautotrophy. So they're doing an autotrophic metabolism using chemicals, inorganic chemicals instead of light. So chemoautotrophy here, whereas in the heterotrophic metabolisms, light penetration doesn't matter. All that matters is oxygen penetration. From this 2 millimeters down, the only type of heterotrophic metabolism that's going to work is anaerobic respiration and fermentation. So these are two types. Anaerobic respiration is a type of respiration, and fermentation is a different type of metabolic activity that can be done in the absence of oxygen. Okay, so which of the following would you predict to find in the bottom layer of a microbial mat where there's no light and no oxygen? Okay, if you said fermenters, you are correct. So chemoautotrophic eukaryotes, although this was a decent guess, we actually don't know of any eukaryotes that are capable of doing chemoautotrophy. So although chemoautotrophy could go on there, there's no eukaryotes that are capable of doing that. So this is not correct. If you said photoautotrophic eukaryotes, this answer is incorrect because you can't be photoautotrophic if there's no light available. So if there's no light available, photoautotrophy will not work. So although there are photoautotrophic eukaryotes, Photoautotrophy will not work. As for aerobic prokaryotes, that will not work simply because there's no oxygen available for any aerobic respiration. So fermenters is the correct answer here. And fermentation is a process, if you can remember back to Bio 140, that does not require oxygen. Fermentation can be done in the absence of oxygen. So why does any of this stuff matter? Why do we care about metabolic diversity at all? So remember that bacteria and archaea, they're crazy, crazy abundant. They are everywhere. And so they're always sort of in the environment in high abundance, doing all of these different types of metabolisms. And some of these metabolisms are absolutely vital for different processes that humans depend on. One specific example is the nitrogen cycle. So this is a sort of simplified schematic of the nitrogen cycle. And this is just one example of the different what's called biogeochemical cycles that bacteria and other microorganisms contribute to. And so basically, the way the nitrogen circle works is nitrogen is typically locked up as nitrogen gas, N2. This is a very inert form of nitrogen that's not biologically reactive. And actually very, very few organisms are able to get access to this N2 gas because it's so non-reactive. However, there are some bacteria and archaea that can do this process called nitrogen fixation, which basically takes this N2 and turns it into a much more reactive form of nitrogen called ammonia. And that ammonia can be further nitrified into nitrates and nitrites. And all of these nitrates, and ammonia are super, super important as organic N in proteins and used for plant growth. So if you think about when you use a fertilizer, nitrogen is a key ingredient in fertilizers, and that nitrogen has to be in a very specific chemical form, typically nitrates, nitrites, and ammonia. If you just put nitrogen gas in, plants will not get access to it. So if we didn't have these bacteria around with these weird, interesting, different metabolic processes, we wouldn't have these different forms of nitrogen to give to plants to allow them to grow. And so these processes, these metabolisms, are extremely important for agriculture, for just one example. And it's also important in other cycles that we won't talk about right now, things like the sulfur cycle, the carbon cycle, etc. And there's a lot of human uses outside of just agriculture for this metabolic diversity. One example is bioremediation. So bioremediation is essentially just using microorganisms to remove contaminants from the environment. And there's lots of different species of bacteria and archaea that we're using for bioremediation. One great example is bacteria that can take a toxic compound, something like petroleum, and use the toxic compounds or the petroleum as an electron acceptor and donor, and can actually has a metabolism that'll take that toxic compound and convert it into CO2 and water. And so what you can see here is this is an area where there's an oil spill. So there's oil slick. This is a petroleum-based sort of hazardous chemical. And what these guys are doing is they're spraying bacteria and a little bit of fertilizer onto these oil spills, and the bacteria will break down that oil slick and the oil that's sort of contaminated this area and convert it into CO2 and water. So it's a way to bioremediate or clean up the area. The other great thing that we can use some of these bacteria for are biofuels, which is basically just fuels that are produced from living organisms. And we can use this basically, we can use these prokaryotic metabolisms, we can capture them, we can use the high energy molecules that those metabolites produce, and you turn those into sustainable energy sources. So instead of extracting oil or non-renewable gases from the ground, things that we know there's limited amounts of these resources on planet Earth, we can replace them with renewable resources like growing cyanobacteria. So cyanobacteria are the green things that are growing in these tubes. The energy that those cyanobacteria can be converted into biofuels that can be used for lots of other sources. And in fact, this is something that's happening right here in Harrisonburg, where the Rockingham Memorial Hospital is actually using methane gas that's produced from a local landfill to generate power for the hospital. So let me just show you that in this video.
I'm Dr. Michio Kaku. As a physicist and a futurist, I believe that reducing carbon dioxide emissions is one of mankind's greatest responsibilities. Chevrolet is investing in projects across America to help reduce CO2 by up to 8 million metric tons over the next few years. We call these projects carbon stories. Americans make over 250 million tons of trash every year. As trash decomposes, it produces gas, partially CO2, but about 50% methane. And that methane builds up over time. So what can we do? Well, we could destroy it by flaring, or we can do something much more exciting.
We receive an average of 300 tons of trash a day. Every bit of trash that's behind us here will be, it'll be sealed into the landfill where it'll start to decompose.
You've got these little microorganisms that essentially eat the trash and produce methane. Methane gas is a powerful greenhouse gas. It's 21 times as potent as CO2 in the atmosphere.
Every day we're producing methane. This doesn't stop producing methane. We capture that material with these gas wells, as you see here behind me, and use that methane to do something beneficial.
The methane gas would get sucked up the well here, down to the main pipe. The pipe comes out of the flare station, shoots down here to the road, runs all the way 3 miles up, goes through the pipe to the hospital.
What we have right here is actually where the landfill gas and the pipeline comes up from the landfill. When it comes to the hospital, we actually burn it in our boilers as a substitute for natural gas or diesel fuel. It generates steam, and then we use it to heat our hot water. We use it to provide heat in the wintertime. We sterilize our surgical instruments with it.
There is a pleasant, intriguing paradox of things that are dead and rotten in the landfill, and normally we can think of as being dirty, is then being transformed into making this to a clean, sterile, healthy place.
It's hard to stand in this community anywhere and look around and not appreciate the environment and want to make sure we're protecting it for generations to come.
Our motto is get well and live well, and so our employees say this is a way that we can also help our community get well and stay well.
It is pretty cool that we have a beneficial use project that's happening at the local hospital where you can actually see it, in action. If you go into the hospital over there, you can say, "Oh, look, it's warm in here." It's because we're burning the methane from the landfill. We're not burning so much fossil fuel.
Chevrolet.
So a pretty ingenious use of these microbial metabolisms that have evolved. So we're going to stop there for now, and we'll pick up with our next lecture talking about how did bacteria evolve this wide range of metabolic pathways. So stay tuned for that next lecture. Talk to you later, cats and kittens.